Okay, is everyone settled and ready? All right. So, hi everyone. Thank you for coming. Today I'm going to be giving my MS thesis defense on low-dimensional signatures of aplasia neuronal population activity revealed by matrix factorization and spatial mapping, or in short, how long-term memory looks like at the neuronal level. So, intuitively, the differences between short-term and long-term memory, one of the main factors is recall time. So this means the time between the incident and when you recall it. So for short term, this can be within a couple of minutes. So if I were to ask all of you, what was the color of the previous slide? You would all say
Teal, white.
teal or white or blue. Yeah, so that's good. So your short term memory, you're going back and recalling that. But if I were to ask you that 24 hours from now, that would be considered long term memory. So if I do ask you that tomorrow, please don't forget about it. And intuitively, we know the differences between short term and long term memory. But in this talk, we want to look at that biologically. Specifically, what is the differences between short-term and long-term memory in the neuronal recruitment and population dynamics. So in order to look at this, we need to look at how memories are formed and stored. So we can do this by looking at how they're changing in the properties of their neurons and their connections, what we also call neuroplasticity. So a neuron looks like this, in which they send electrical and chemical signals across the neuron to other neurons in the body. And this helps us do everything we know from breathing, talking, thinking, and learning. And in the human brain, there are 84 billion or estimated 84 billion neurons, and they work together kind of like a symphony, in which they help what we do over time and kind of like this image. So how can we see this neuroplasticity in action? Well, we need a simple form of learning to do so called operant conditioning. And operant conditioning is showing how behaviors are controlled by consequences. And this theory was originated by Thorndike in 1911. And in this theory, there is sorry, this puzzle box experiment is where a starved cat is in a box with a latch, and what they have to do is learn to open the latch to get out and get their food. And what he saw was that after repeated trials, the cat was getting out faster. And we want to see that in the brain, the neurons indicated by the circles here, are they acting differently or the same between short-term and long-term memory of this operant conditioning paradigm? However, the human brain has billions of neurons, so we need to look at, which makes it difficult to see the patterns. So we need to look at a small brain model, and for that we can use an aplasia. You may be wondering, what's an aplasia? And an aplasia is a sea slug, and this over here, the head is facing towards me. And in their entire central nervous system, they have around 20,000 neurons. Specifically, we can look at the buccal ganglia or the feeding circuit to look at the effects of operant conditioning. And the nice thing about buccal ganglia, and there's like around 2,000 neurons, but also we can image these neurons since they're really large and identifiable. In this image alone, there's around 124 different neurons that we can look at and see the activity from. So how does operant conditioning look like in aplasia? Well, in aplasia, they have two different options when they're eating seaweed. They can either fully ingest or eat the seaweed, or they can reject the seaweed. And over time, what we've seen in operant conditioning is that they have more of an association that if they eat the seaweed, they will continue to ingest it faster. And this happens in the real life animal. But even if we dissect out the buccal ganglia, we can still have these operant conditioning features in vitro, so outside of the body. And we can look at the subset of neurons within the buccal ganglia. Buccal ganglia is central pattern generator, and the CPG produces rhythmic and patterned outputs called fictive buccal motor patterns. Fictive because they're not in the real life animal. So we still have the similar association of the buccal ganglia associating reward with certain patterns. So now we can look at how we classify these fictive buccal motor patterns. So buccal motor patterns, or BMPs, can be classified in one of two ways, either an ingestion pattern, an IBMP, or a rejection pattern. And we can classify this based off of the nerve activity. So on the left-hand side, what we have, okay, so look at this thing. So in the real life animal, the equivalent is going to be, I'm going to use an example with my water bottle. So it happens in two phases, this pattern. You have protraction, this would be me going out and reaching my water bottle, and retraction would be me bringing it back into my body, and ingestion is me drinking the water. Where your hand closing around the water bottle is the closure feature, so in purple. And different nerves on the right-hand side that you can see, they are active during different phases. So buccal nerve 1 is active during protraction, whereas 2 and 3 are active during retraction. But the key nerve to note is that the radula nerve in purple, if it's more active during retraction than protraction, then we consider it an IBMP or ingestion pattern. Whereas the opposite is true. If the radula nerve is more active during protraction than retraction, then it's a rejection pattern. And we can look specifically at the esophageal nerve that's up here and to stimulate or to use in our operant conditioning paradigm for the aplysia. So now let's look at what that looks like. So Yutomo Mohara, a postdoc in the burn lab, is training these isolated buccal ganglia. So how does he do this? With the first preparation that we have that we've isolated, this is called the contingent. Following every ingestion pattern, we note the time period that the ingestion pattern ended, and then we're going to stimulate the esophageal nerve, which releases dopamine across the ganglia. So this way, the preparation is contingent upon receiving dopamine following an ingestion pattern. So we want to make sure that this association is happening because of the dopamine with the ingestion. So we need to have a control or a yolk preparation that we're still going to stimulate at the same time points we gave to the contingent, regardless of the behavior that the yolk is presenting. So you can see the ingestion patterns are happening different time points, but we're still stimulating at the same time that the contingent is stimulated. So this would be one pair of ganglia, one contingent with one yolk. And another pair looks like this, in which you can see the contingent times are the ones denoting when we're getting stimulated. And in total, we have 11 pairs of these isolated ganglia, or 22 ganglia. Okay, so after training, how do we image and analyze to understand more of what's going on? Well, we do this for long-term memory in 3 main steps. The first step, which we just talked about, is the operant conditioning training. And the key part to note between short-term and long-term memory is that 24-hour resting period after training, which we do, and to stimulate the long-term. And then after that 24-hour rest, we have the voltage-sensitive dye recording, which is capturing our neuronal firing activity. And then after this, the third step is the data analysis to find underlying patterns from our data to better understand what's going on. So Nick Gonzalez, a research assistant in the burn lab, worked on doing the second step. So what he was able to do from the nerves is classify these different patterns. And in this image, you can see the contingent part of the pair, for one example, has 3 ingestion patterns in green, whereas its yolk pair had 0 ingestion patterns. So we wanted to see across all of our 11 pairs, the fraction of ingestion patterns. And on the left-hand side in green is our contingent, and the right is our yolk, where each gray line is denoting one pair. So in this top pair we have here, this one dot right here, this means that the contingent ganglia had all ingestion patterns, whereas its pair down here had a fraction of IBMPs of 0.75. Yeah. So we did this for all 11 pairs, and we saw that the contingent had more ingestion patterns than yolk. So this means that the operant conditioning is producing more ingestion patterns. That's good. And what Nick was doing was recording So Nick is going to be recording the ganglia. So what Nick does is we have the ganglia preparation, and we stain the ganglia with voltage sensitive dye. And this captures the change in membrane potential, or how much the neuron is firing. So when Nick does this, on the left-hand side, we're showing the top 35 most active neurons. And on the right-hand side, we can see the activity. So from the top, remember, those were the nerve-based classifications. And for behavior, it's straightforward how we're going to classify them. It's either an ingestion or rejection pattern. However, the neuronal activity is pretty complex, and it's not as interpretable, so we need to use a new method in order to understand what's going on in both the neurons and time patterns. So this is the part of the work that I was focusing on, in the using non-negative matrix factorization to capture the neuronal and temporal patterns. So we use NMF because of that non-negativity constraint, which makes it more interpretable for us to use because we have neuronal data. There's either firing or no firing. but also time, you don't go into the negative realm. So we use NMF. And what NMF aims to do is reconstruct X, our original data matrix, which showing neurons firing over time, into a product of N and T, which are vectors in this case. That's where that dimensionality reduction comes into play. And what it does is it starts off by initially, So what N and T are trying to do is N is trying to capture the neuronal patterns or how much each neuron is contributing to the overall firing activity. So in this case, this first one is not contributing where the second neuron is. Conversely, on the top, we have our temporal pattern. So this shows the frequency and intensity of the overall pattern. So in this case, we have 1, 2, 3 patterns that are going on in the entire time. So what NMF does is it starts off with initially randomizing N and T, and then it'll hold N fixed, update T, then it'll hold T fixed and update N. That's one iteration to calculate the error. And then it'll keep going through this process or many iterations, either for a max of 100 iterations or when it reaches like a local minima or small enough error. So this is what we do. And from this, it's not always going to be as simple, like very easy to take out the patterns, as you can see from this toy example. But in our data, so in the middle, let's break it down of what we were doing. In the middle, we have our binary spike times, and we're convolving them with a Gaussian to go into the continuous realm. So that's in the middle where white is going to be more firing of the neurons and black is going to be no firing. And what NMF outputs for us is on the top is going to be our temporal patterns. So this shows us there's 1, 2, 3, 4 patterns going on in this case. And then on the right-hand side, we can see the neuronal patterns where this indicates that this top neuron is more contributing to the protraction module than retraction, and vice versa for this bottom one. And on the top, we can see the patterns, but we can validate it using that nerve-defined time steps that Nick produced, and we can see the orange is still going to be protraction followed by retraction. We can see they line up pretty well of capturing these time patterns. So let's dive a little bit more into the temporal realm. In order to compare our different time patterns, we know that protraction and retraction can be varying from pattern to pattern, but also the entire duration can be varying. So we need to normalize our protraction phase and normalize our retraction phase so that we can compare across our data. So what we do after normalizing is we go into the space where protraction is now from 0 to 0.5, and then retraction is from 0.5 to 1. I can see that dashed line is the transition point between the two phases.
Now the Gaussian pattern that you currently use have the same variance across the whole time period?
Yeah. So then this is the dashed line that we want to have for the transition. So we can look at it in one of the examples. So on the left-hand side, you can see those transition points are in the dashed lines. You can see they're varying. They're not always at 0.5. And those curves in orange and blue are the NMF outputs. And then after normalizing, we can see that all of the dashed lines are in the middle. So that's good. We've been able to normalize, and now we can compare across different BMPs. But another varying part that we have is per experiment, there's a different total number of these patterns. So one, in this case, could have 4 or 5, and another, there could only be 2 BMPs. So what we do is we average all of the protraction modules and all the retraction so that each experiment has 1 orange line followed by 1 blue line from our NMF output. And when we do this across all 11 of our pairs of contingent and yoke, this is the breakdown. So on the left-hand side in orange, these are protraction phases, where on the top is that contingent preparation, on the bottom is our yoke control. And then similarly for the blue on the right-hand side. And what the circles denote is like for contingent preparation number 6, this is the peak point or the highest point of the curve. And from this, we can't see any obvious patterns. So what we wanted to do is average each of these 4 groups. So now we're going to get 4 different lines. And when we do this, we can see some more notable differences. So on the left-hand side in the solid orange, we have our contingent average, when on our dashed orange, we have our yoke average, similar for the retraction module. And something to note is a little easier to see visually from this example or from the average is that our time to peak for the contingent retraction module is earlier in time compared to our yoke average. So this was something that we wanted to look further into and compare across all of those 11 pairs. And when we did do this between our protract for both modules, we looked at that peak time point between the contingent and yoke pairs, and we did see that in fact the retraction module, the peak time point is earlier in time in contingent compared to yoke, and this wasn't the case for the protraction module. Another piece that we looked at was how high are the magnitudes of these different curves to see if there were any differences in either module, and we didn't find any differences between either contingent yoke and protraction nor in retraction. So that was something to note. And then we compared, this is our long-term operant conditioning results, and now we wanted to compare them with short-term results from a previous study by Renan Costa, and former member of our lab. And on the left-hand side, we can see the short-term retraction results. So that same type of contingent yoke pair where each of the dashed lines across are going to be each pair. And we can see that he also found that the contingent was happening earlier in time for the peak time points, whereas, and we also have that in ours. And what he's looking at for the pre and post on the bottom, pre means before operant conditioning, post means after. So we can still see that the temporal shift and the peak points look similar to what ours are doing comparatively. So that's one point. We found that the temporal signature between short-term and long-term memory are similar in this operant conditioning paradigm. So now that we have that, we've looked at our temporal patterns on the top and we've compared that to short-term. Now let's go look at the neuronal patterns on the right-hand side here. Recall that this line stem plot shows that this neuron on the top is contributing more to protraction than it is retraction. And similar, this neuron on the bottom is going to be contributing more to retraction than protraction. And in order for us to compare, again, we need to do some normalization. So the most contributing neuron, we're going to normalize from 0 to 1, where the most contributing neuron is going to be that 1 per module, the top value. Oh, and what we need to do is we want to maximize the number of non-overlapping neurons. So we want to find neurons that are either contributing to protraction or retraction. So we set a threshold which maximizes this at 0.4. So now we're only looking at neurons that are contributing to either protraction or retraction from 0.4 to 1 in that realm. And when we show these overlapping neurons, on the left-hand side, again, we still have our protraction, contingent on the top, yoke on the bottom. Similar for the right-hand side for retraction. What we can notice is that on the top right, or where my marker is, for contingent protraction, there seems to be more neurons active in the top right-hand corner compared to yoke protraction, but this trend is much more pronounced in contingent retraction compared to yoke retraction, just the sheer number of neurons that are contributing to these patterns. So we wanted to look at the percentage of neurons per experiment that are above 0.4. And when we did this for retraction, we did indeed find that there is a higher percentage of these contingent neurons. Yeah, contingent neurons that are affecting to retraction. And then I want to sum up the total contribution values to see if not only are there more neurons, but are they contributing more to the overall firing activity. And we did indeed see that this retraction module is firing and contributing more to the overall patterns. And this is not the case in the protraction module. So similarly from before, we want to compare these long-term results to our short-term. And I reran this analysis on running the short-term operant conditioning data. And on the top, we can see short-term, in which there are no differences between the percentage of neurons between protraction and retraction modules. Whereas in ours, we saw a higher percentage of neurons being active in the retraction contingent module. So now we've seen that, okay, temporal signature was the same, but the neuronal signature is different in long-term versus short-term. So we did see that spatially, or from this spatial area, we can see that a lot of the neurons, it matters where they are and the boundaries are drawn. So this is currently hand drawn by Nick, which takes hours to do. So we wanted to look at another way to automate this process. and to make it faster. So Pranav, sitting here, is a undergrad at Rice, and I'm a mentor of him in this project. And what he's been working on is automating this neuron drawing process using the segment anything model from Meta. So on the left-hand side, we can see the original ganglia or picture that we want to look at. And after 3 or 4 minutes, the SAM output on the right-hand side is showing us the different boundaries that it has. And on the bottom, these are the true segments that Nick had drawn. And in comparison, the SAM segments are still drawn pretty well. They're non-overlapping and they're in the same spatial areas that Nick represented them in, so that's good. There still is some noise on the bottom, which we're currently fine-tuning the model for, and it's not picking up every single neuron that it's supposed to. But we're still working on this and hopefully going to make it a website so that other scientists with similar types of data to ours can use it to make their annotation process go faster. All right, so in conclusion, what we've seen from this study is that NMF revealed an increase in the recruitment of retraction neurons in long-term operant conditioning. We saw that from short-term to long-term, we had similar temporal signatures. However, we had different neuronal patterns and that increase in the retraction contingent percentage, but also in total amount of the neuron values. So we've seen this neuroplasticity in action with our study, and now the future directions of this work would be to see to what extent these conclusions are relevant to vertebrates and like in zebrafish with other fictive motor patterns that can be looked at. Okay, so now I'd like to thank the Burn Lab for bringing me onto this project and working with me this entire time. And we're currently writing a paper to submit into the Journal of Neuroscience. I also want to thank the Ajeng Lab and Rory for listening to this presentation practice many, many times and helping me through the entire graduate school process, and also to my parents and my friends at soccer who have been along with me on this entire 2-year journey so far and many more to come. So I'll be taking questions now. Thank you.
You want to go with the committee first? Basically, the tradition is that first you ask questions and then you possibly ask the guests to leave the room, and then for Sara to stay back to get ready to drill. But that depends on the committee. Any questions from guests and committee members?
Sarah, can you help me understand
Yes.
the pairing process?
Yes.
And then how many ecclesia were involved in collecting that data?
For sure. Okay, I'm gonna go back in the beginning. A good question. Okay. Now it might be easier without this. Okay, so the whole process of it, or how they're paired, or which specific part?
So the whisker plots that you showed, which were really important in understanding the time difference.
Yeah.
There wasn't a lot of data there, but what's behind that? How many ecclesia?
Okay, yeah. So
how do you pair?
Right. So there's 11 of these lines, and there's going to be on each side is 1. So there's 22 total ganglia that we're using, so 11 pairs.
How many ecclesia?
22.
22.
Yeah.
Okay.
Does that make sense?
So you don't use they're bilateral, but you're just looking at one.
Just one. Yeah. We're looking at one.
Okay.
Yeah. And like when we're imaging the left side of one.
Okay.
Yeah.
And the pairing?
The pairing is based off of the training that we did. So this was before that 24-hour period. So recall how here the timings were all based off of the contingent, right? So this first preparation on the top, every time it finished, we put down like, okay, at 2 seconds, the first one finished an ingestion pattern. And then the next experiment that I'm doing with the second ecclesia, at 2 seconds, I'm going to still stimulate it because that was based off of our contingent. Does that make sense?
Thank you.
Yeah. Good question. Yeah.
So you look at one side of the ganglion.
Yes.
What do you anticipate would happen if you looked at the other side of the ganglion?
So what I've learned, that's a good question. Okay. I have the picture of that. So they're supposed to be symmetrical in what they're doing, but I've learned that some of the neurons for protraction and retraction that are like specifically contributing. Some of them are on one side and not the other, so it's not a direct map of that. And I think if that map gets built, then it'd be easier to see the symmetry across the left side and the right side.
Actually, I was talking about the top and the bottom.
Oh,
So there's an underside there that we're not.
oh, yeah, the other side of it
The other
too.
side.
Maybe it shows more. Maybe it'd be like, we can do that in an experiment.
Future directions.
Yeah, future direction. Yeah, that'd be a nice area. Nick, are you ready to image that side?
The right side.
Oh, okay. Yeah. I'll get back to you in a couple months.
What is the imaging depth of your optics there?
I know we have, okay, so like the ganglia is flat on the petri dish, on the dish, and then we do it from we're only looking at one of the angles, but when we image, we have a high left plane, the low left plane, the high right plane, and the low right plane. But the specific depth, I would look to Nick to answer that question, as he's the one who's doing the experiment.
So I positioned the camera at the focal plane that captured the most neurons.
Yeah.
So
That's what like we're using the high left plane because it captures more of the neurons. But when we go to a lower plane, it becomes like distorted on the top because it's so close that it can't even focus on those neurons. So you only get just the outside ones.
How many neurons of this 100, 200 neurons can you say at once?
It ranges from, I know the smallest is like 33 to 124 is our highest, but on average it's like 78 in this data set. Yeah.
Some of them are relatively active. Go to the.
Yeah, even
Go to
yeah.
the report there.
This one? Yeah, I mean some of them are not as active. I mean these are the
Like if you look at neuron 36, it's shows very little activity.
well, these are showing the most.
these are the most active, yeah. I
neurons,
can
You can see in the image there are many neurons where there's not a colorful circle around it. Well, I checked those and those were not showing significant activity, which is why they're not included here in this in these traces.
Now 36 is in the left picture also.
Should
I
be.
guess hard to see.
37. Is it?
In the middle.
Oh yeah,
A middle,
it's
yeah.
right there.
Even though it's not active in that right side, but it still shows up in the circle.
Yeah, there's still some firing.
So can you give me more like justification on your normalization? So particularly, the question, maybe try to counter argue that I would think the precise tempo sequences of the spiking is the most important information the neuron gives.
Yeah.
And your normalization actually normalize the time scale.
Yeah.
And then you look at when it peaks,
Peaks,
right?
yeah.
So then it can possibly like you see a change of the peak time because
They're normalized.
they're normalized. And why this is a more accurate measure for the peak time rather than not touch anything about the time scale.
Yeah, I think the main reason why, even when looking at the just like original data, let's say in this example, the protraction starts and then retraction happens, right? But this transition point for the first one, it's like still at the start time of protraction. And if we were to compare based off the original nerve timings, like I think it would be, or from what I'm understanding of it, I think it would be significantly far off, if it makes sense. Like it would be more inaccurate. It would say like, oh, protraction for this first experiment peaks during protraction.
But you could center it on the
Yeah,
transition point. So
that's true. Yeah.
Yeah, like that's exactly what I was asking, right? If you just align to the transition point, then you have absolute measure of the time that you did not change its scale from trial to trial.
That's true.
So I think this example is a good illustration here. Like you could have basically peaked at exactly, if you look at the orange, right? You could have peaked at exactly the same absolute time, but because the first one is so much shorter, when you normalize, you're going to see a change.
Stretches.
Yeah, you're going to see a change in the peak time.
Yeah. That could be something I could try and with it.
But I
I
think
don't-
fundamentally I'm interested why the normalized one would be a better method than not normalized.
Yeah, so this is more, it was more of a decision based off of the short-term experiments too. So we're trying to compare them between short-term and long-term. So the same methodology that we're using in this was developed by Ren and Costa as well, where that similar type of normalization from protraction being from 0 to 0.5 and 0.5 to 1 for retraction was based off of. Like it'd be more accurate to compare this normalization method with short-term results than if we were to, like I understand what you're saying, but I don't think it would be as accurate to compare our results with short-term in that sense.
But I still do not get why. But I think
Yeah.
we could talk more.
Yeah, we can talk about it
that.
later. I can try it. Then. What do you mean?
You look at the rise time to show that
Right.
all signatures are equivalent.
Mhm.
So I'm curious if you envision a way where if you didn't normalize, you could still make conclusions there, or maybe that would
Some
be one
other-
advantage.
Yeah, then maybe we'd look at like time to half peak. That was some other areas that we looked at, not just like the peak time point, but the time before and after to half peak because it was more comparable. Maybe it might fix the normalization problem too, or area. I think. I don't know.
So about the non-negative back matrix vector.
Yeah.
Your input is continuous time trace, but your analysis on what this algorithm is doing is purely discrete. Is there a good way to understand why the outputs time traces are continuous?
Wait, could you explain that again? Sorry.
I mean, your time traces that you put in, these are continuous time impulses.
Yeah.
But the decomposition that comes out is also continuous. But yet the decomposition itself is like a discrete factorization. Why should I expect any continuity in my output trace? I mean, it seems rather remarkable.
Well, like temporally it makes sense, right? Like why would it be discrete for the time traces? Wait, do you mean the time
I
traces?
don't see any that's necessary relation in why the decomposition itself should be continuous. Right there.
This part of it?
Or the next.
That's what this.
Why are they smooth? Why should the orange and blue curves be smooth?
Well, like even in the fire, like if I'm understanding it correctly, like in the behavioral patterns.
I understand biologically why that should,
Oh, yeah.
but why should that come out of the algorithm?
Because the algorithm works that way.
How'd you go from spiking data to smooth Gaussian looking curves?
Yeah, it was a convolution with 1 second bins. The big
It's the
option.
Gaussian application, or treasure question. So this is a Gaussian filter put on the spikes.
Oh, sorry, I thought I explained that.
Was that Gaussian filter put on the spikes because the spike data is consistent or?
It was just because that was from a binary domain, and to go into like the top part of the patterns, then it's more interpretable with like how much it's changing over time with these curves instead of just time points. Yeah.
So the voltage stain that you're using, does that capture individual spiking events or?
Yeah.
Oh,
Yeah.
Show,
We have the
put that up there.
and this one, yeah, we have like
Point out some spikes.
yeah, like so these ones are spikes. We have little red dots in our system that'll show you like this is a spike. And these parameters that Nick built it around, we're applying it to all of our data. So it's the same spike parameters to identify a spike in all of the
The
data.
spikes are the downward deflection,
Yeah, these like little
and there's a Gaussian filter put on those.
yeah. So it went from spike time, like it literally would say like, oh, at time 3 there was a spike, time 5 there's a spike, and then we go to this continuous.
so the Gaussian spectrum, the convolution is kind of pulling out the bursting activity.
Yeah, exactly.
Bridging them together, so you.
Use NMF for it. Thank you.
So how did you decide the kernel to be 1 second?
Yeah, so this is also based off of what Renan was doing. What was told or what I read was that it relates to in vivo patterns or in the real life animal where these patterns are happening over tens of seconds. So it's to match what the in real life patterns are happening. Yeah. Yeah, I know, I also was like, why'd you do that?
Why NMF and not some other dimensional reduction?
Yeah,
algorithm?
exactly, that's a good question. So not only is the non-negativity constraint what we need for this type of data where it's neuron spiking and that temporal part, but also we don't use PCA, or why we do want to use NMF is because it gives you parts of a whole or parts of the actual data, original data that we have. So it pulls current underlying patterns from the actual data. It's not just giving you a principal component, which PCA would do, and that would be very uninterpretable for our data. So that's why we use NMF. And it was also used in short-term operant conditioning as well, which we talked about.
And if is pretty standard for
Yeah.
the space, right?
Mm-hmm. There are other types of it too, like sequential NMF, where it tries to show you which neurons are talking to which. Like Macavicius talked about that, which would be, yeah, interesting.
Any other questions? If not, can we please ask the guests to leave and now, does the committee want Sara to stay back for more questioning, or are you done with questioning?